Hello everyone, my name is Melanie Avila and I'm presenting the work titled Comparative Genomics and Structural Analysis of Limosilactobacillus reuteri Gene Regulation for Glycerol-Utilization. In bacteria, glycerol is metabolized to 3-hydroxypropionaldehyde and 1,3-propandiol. Both of these compounds are precursors of chemicals used in the food and cosmetic industry. Limosilactobacillus reuteri produces both of these compounds in a safe way as it is non-pathogenic, and this is the reason we will be studying it in this work. Our first goal was to perform a comparative analysis of the genomes of Limosilactobacillus reuteri and microorganisms that use glycerol to generate 1,3-propandiol and 3-hydroxypropionaldehyde. And our second goal was to obtain by homology modeling the molecular structure of the regulatory protein POCR identified in the PDU operon of Limosilactobacillus reuteri, related to the production of the compounds previously mentioned. So, in order to begin with our comparative genomic analysis, we began by seeking query amino acid sequences of all the components of PDU operon, described for another bacteria called Secundilactobacillus collinoides. Once we got all the proteins we could find, we used BLAST tool to find the position of the proteins in the genomes of Limosilactobacillus reuteri, Klebsiella pneumoniae, and Salmonella typhimurium. After knowing the position of every protein, we built a comparative genomic map for the three bacteria. For the homology modeling, we began by using Swiss model with query sequences and templates obtained by PDB. We obtained structural models of different domains of POCR protein. With this set of structures, by performing structural alignment, we obtained a complete POCR model using the program STAMP. Once we had the complete POCR protein and using the program SARA structure with level of calculus MNDO AM1 PM3 and the field force Jamber 3, we obtained the enhanced model of POCR. And by using the molecular dynamic tools that SARA offers, we obtained a refined model of POCR. Finally, using PDB-SUM, we checked the quality of the model with ProCheck and the DNA interactions with NOPLOT. First, we have the results of the comparative genomic analysis performed. In these graphics, we have the gene organization of PDU operon in Limosilactobacillus reuteri, Klebsiella pneumoniae, and Salmonella typhimurium. As you can see, they all have the same organization of genes from PDU A until PDU H. After this, Klebsiella and Salmonella have some additional proteins, but they maintain a similar organization with Limosilactobacillus reuteri. Also, we can see that Salmonella and Limosilactobacillus both have the regulatory protein POCR, which goes in reverse direction comparing with the other proteins of the operon. For the homology modeling, from Swiss model we obtained that POCR has two domains, a cobalamin binding domain and a DNA binding domain. After structural alignment of both domains using the program STAMP, we obtained a complete POCR protein. Using JASARA, we obtained the enhanced and refined model of the protein and its interactions with DNA and cobalamin in its binding domains. After checking the quality of the model with the program PDB-SUM, we obtained that most of the amino acids of the model protein were in the red area, which is the favored area. This means that the distribution of the torsion angles in the protein structure is good. As the first conclusion we have is that Limosilactobacillus reuteri has similar gene organization for the PDU operon as Salmonella typhimurium and Klebsiella pneumoniae. The regulation of the PDU operon could be mediated by the POCR protein. Also, POCR has a DNA binding domain and a cobalamin binding domain in its structure. These domains could be the ones related to the regulation of PDU operon when using glycerol as carbon source, as glycerol dehydratase is a cobalamin-dependent enzyme. Finally, I want to thank the Vice Rector Office for Research and Postgraduate Studies from la Universidad Nacional Mayor de San Marcos for funding this study. Thank you.